My name is Darren Ennis. I'm a translational research scientist in the Department of Surgery and Cancer at Imperial College in London. So as a translational scientist, I get a unique opportunity to work with many different people and many different collaborators within Imperial College. My expertise places me at the intersection between the NHS and the university environments. I work mainly on human tissue samples. In our case, ovarian cancer tissue, mainly from patients that have been newly diagnosed or those that have relapsed. Why is this relevant? The cancer translational scientist, we play a crucial role bridging the gap between laboratory research and clinical application. We translate these findings from basic science into practical treatments, ensuring innovative therapies reach patients more quickly.
Hi, my name's Katie Tyson. I'm a lab manager and an in vivo study coordinator for the Department of Surgery and Cancer at Imperial College. We work on ovarian cancer. Ovarian cancer is also known as the silent killer because unfortunately most women are diagnosed at stage 4. At this point the disease has already spread throughout the peritoneum and it's then very, very hard to treat. So yes, I work with mouse models. A little black mouse called C57, and these mice have a cell line which has been developed to be used on this strain of mouse called the ID8 cell line. And we can edit this cell line to knock out genes representative of human disease. So I'm sure a lot of people would have heard of the BRCA gene. We have deleted the BRCA gene from this ID8 cell line so that we can see how a loss of the gene can affect the disease. So a lot of our research focuses on the tumour microenvironment, and this is the environment that is directly adjacent to the tumour within the body. Ovarian cancer is able to grow and spread so well because of how it interacts with our own immune systems. We have a cell called a macrophage, and ovarian cancer cells trick these macrophages into thinking that they are not dangerous, and therefore they use them to promote their own growth and they disseminate throughout the abdomen. So we study the different knockouts that I mentioned before, such as the BRCA1s and the BRCA2s, and we treat them with different drugs to see how different gene knockouts can introduce sensitivity or resistivity to chemotherapy drugs. So in these cases, we will inject the mice with cancer cell line, such as the BRCA1, and then we will treat them with a drug called cisplatin, which is in use in hospitals, and we will study how this changes the morphology of the tumour, how quickly the tumour will shrink, and also we do this to test how the drugs interact with other tissues in the body to make sure that it's not going to be toxic in a human. So my colleague Darren is focused on the human side of the lab, and we collaborate together using his immortalised cell lines in humanised mouse models. Humanised mice are mice which have an impaired or no immune system at all, and that means that they don't reject human cancer cells when injected. This allows us to study how the human cancer will grow in a living system.
So overall, I don't really use animal models. I tend to use human tissue direct from patients. But on occasion, we do manage to find primary patient cell lines that have spontaneously immortalised. What does that mean? It means that sometimes cells escape the normal mechanisms for growing and dividing, and they basically grow indefinitely. This is a great resource for scientists, as they have cells that are able to grow in a reproducible manner. We can then put these immortalised cells back into animals, and then we can see how they would grow as they would grow in humans. This allows us to examine and study cancer in a complex environment, which we cannot really do in a tissue culture dish. So while advancements in translational science have really increased our understanding of cancer and improved therapeutic approaches, it is unlikely that translational cancer science can completely replace animal research. For the moment anyways. The main reason for that is that cancer is a very complicated disease, and that impacts the whole body and not just one part. It involves many different cells, tissues and organs. So studying animals helps us see how cancer grows and changes in real living body, which we can't really do by looking down cells in a cell culture dish. But that's kind of changing rapidly, especially with the advent of things like organoids, which are kind of miniaturized and smaller versions of organs. We can now really study disease processes in cancer, bringing us one step closer to minimizing the use of animals in research.
So I think Getting word out there about animal research is very important. I think everyone has this sort of idea of you spraying perfume in rabbits' eyes, but actually in the UK we are very, very regulated in what we do. A main part of my job is to go through each study application and ensure that everything lines up with what's allowed within the licence limits. Each animal is protected and we do the utmost to ensure that they don't suffer.